Today I'm answering this Edexcel Biology 9-1 GCSE paper, and it's a specimen paper, paper 1, and we're going to be doing a complete exam walkthrough, so I'll be writing all the answers in as well. Question 1. Figure 1 shows the times when Homo sapiens and some of their ancestral species are thought to have lived. So you have Homo sapiens, which are the most recent ones here, and then before them came Homo erectus, Homo habilis, Australopithecus afarensis and Ardipithecus ramidus. Fossil remains of Ardipithecus ramidus were discovered in Ethiopia. Describe the evidence that scientists might have used to show that Ardipithecus ramidus inhabited the earth earlier than Homo habilis. So yeah, this is current day, so we are Homo sapiens and we're looking at Ardipithecus ramidus and trying to decide how scientists realised that they might have been around earlier. And how do you actually find out evidence about these things? Well, by finding their fossils. And it's important to notice that older fossils will be found in deeper bedrock, because you find that newer rock obviously is deposited on top of older rock. So first of all, say that you would find Ardipithecus ramidus fossils would be found deeper in the rock layer compared with Homo sapiens. And you need to give a second separate point for the second mark. So we tend to think that the newer species are more advanced in every single way, and in order to be more advanced they need larger brains. So you could actually compare their skull sizes, or if we're being fancy we could say their cranial capacities. Question 2. Suggest an explanation for the extinction of Homo habilis. So what might have caused all of these Homo habilis individuals to have died out? Well, the most likely explanation here is due to competition. They were simply outcompeted by other Homo species, so potentially Homo erectus, came along, took all their food, took all their resources, and effectively outcompeted Homo habilis. So Homo habilis was most likely outcompeted for resources by Homo erectus. There wasn't enough food for Homo habilis. And another thing Homo erectus could have done is introduce new diseases. So quite a waffly answer here. And we're looking for the extinction, so that's why I'm saying which killed Homo habilis. Part 3. Figure 2 shows two stone tools, one used by Homo habilis and one used by Homo erectus. So this one on the right looks more like an arrowhead, it looks sharper, it's pointier. This basically looks like just a kind of generic lump of rock that's had a few pieces shaved away from it. Explain which stone tool was most likely used by Homo erectus. Use information from Figure 1 and Figure 2. So I've already said that the B stone tool looks more complex. If you have a look at Figure 1, you can see that Homo erectus is one of the more modern ancestral species. So because of that, we'd expect them to have larger brains and therefore responsible for making more complex tools. So our answer needs to incorporate all of these points. So start by saying that you think it is stone tool B is most likely to have been used by Homo erectus because it is more complex and we would expect Homo erectus to have larger brains due to their living far more recently. B. The population of humans on Earth has increased significantly, leading to food shortages. The growth of drought-resistant crop plants could lead to an increase in food supply. Describe how drought-resistant crop plants can be produced. So this is a kind of selective breeding, natural selection type of answer. So first of all, you need to state that there is genetic variation due to mutation leading to some crop plants being drought resistant. these drought-resistant crops are selected and are cross-pollinated, so basically you're forcing them to interbreed. Then select their seeds and obviously grow these seeds under drought conditions, because you're trying to increase how resistant to drought they are. Crucially, with selective breeding, you've got to repeat this process for several generations. Two, Streptococcus pyogenes is a bacterium that causes communicable infections. Scientists tested the ability of 2 antiseptics to kill Streptococcus pyogenes bacteria. They spread Streptococcus pyogenes bacteria on 2 agar jelly plates and placed a small disc of filter paper containing antiseptic in the centre of each dish. Figure 3 shows the results of the test after 24 hours of incubation. So the dark grey is the bacterial growth, and then you can see the middle shade of grey is a zone of inhibition, so where the bacteria aren't able to grow, and that's due to the presence of the antiseptic. The larger the zone of inhibition, the more effective the antiseptic is. So just as I look at it, I can see that antiseptic 1 is better than antiseptic 2. Anyway, we're being asked in question 1, calculate the area of the zone of inhibition for antiseptic 2. Give the answer to 3 significant figures, don't forget that. So really what you want to do, they've shown you how to do it for question 1 in case you don't know how to calculate the area of a circle, but I'm really hoping if you're sitting GCSE maths that you do, because it's kind of basic maths you must know about. So what you want to do first of all is measure the radius of the circle. So measure from here to here, and then the equation for calculating area is pi r squared. So we've been given pi, you'll have a calculator, but let's just put pi as 3.14, they've told us that there, times r squared, which is in millimetres, be careful, it needs to be in millimetres, so that's 10 squared. And then when you pop that into your calculator, you get a value which is 314 millimetres to 3 sig fig. And it's crucial you give it in 3 sig fig to avoid losing a mark. Explain which antiseptic is most effective. I've accidentally already done that. If you look up, you can see that the area is much larger for antiseptic 1 in terms of the size of the zone of inhibition. So antiseptic 1 is the better antiseptic, and the reason here is obviously that more of the Streptococcus pyogenes has been killed, hence the zone of inhibition. That's where that bacteria cannot grow. Three, after the bacteria was spread on the plates, both plates were incubated for 24 hours at 37 degrees Celsius. Give a reason why the plates were incubated at 37 degrees Celsius. Hopefully you recognise this as being body temperature. We know that this is a communicable infection, which means it grows within our bodies. Our body temperature is 37 degrees, which must therefore be a pretty optimum temperature for this bacterium. And effectively we want to encourage the growth of the bacterium, so we can see which antiseptic is better. I've kind of waffled on a lot there. You can just write here 37 degrees provides the optimum temperature for the bacteria to grow. The wire loop used to spread bacteria on the agar plate was heated in a Bunsen burner flame before being used. Explain why this aseptic precaution was used. Basically, you want to heat it in that Bunsen burner flame to kill any other microorganisms which could interfere with our experiment. And then this will ensure that only the desired bacteria, so the Streptococcus pyogenes bacteria, are transferred to the loop and no unwanted pathogens are spread on the agar plate. So this method is used to kill other microorganisms on the loop to ensure that only Streptococcus pyogenes, I hope I spelled that right, is transferred to the agar jelly. The ratio of waste to hit measurements can be used to determine the risk of a person developing cardiovascular disease. Calculate the waist to hip ratio for a person with a waist measurement of 830 millimeters and a hip measurement of 0.99 meters. Give your answer to 2 decimal places. Let's be very careful with units here. So let's first of all start by putting the numbers in the right place. And we need to be very careful with units here. So we know that the waist measurement is 830 millimeters. The hip is 0.99 meters, and you need these both to be in the same unit. It doesn't matter if you convert millimeters to meters or meters to millimeters, but make sure you're picking the same unit. So I'm going to convert 830 millimeters to meters, and the way in which I do that is by timesing by 10 to the minus 2. Our 0.99 stays as it is, and then once I've popped that into my calculator, I know I'm doing 8.3 to 0.99 as a ratio, and then in order to get a ratio, just pick the smallest number and divide both sides by that, so that's 0.99, and when you do that you get a ratio of 1 to 8.38. B. Dieting can reduce the effects of cardiovascular disease. Which statement gives a reason why dieting can be used to reduce weight in obese people? So let's have a look. What is dieting? Well, it's when you reduce the amount of calories you take in, and that's how you lead to a reduction in weight. So which of these answers means that? Dieting increases metabolism and growth rate? No. B. Dieting reduces your energy consumption? Yes, that's what I've just alluded to. C. Dieting decreases metabolism. If it did that, then you'd become larger as a result of dieting. D. Dieting increases energy consumption. No, you're taking in less food, so that wouldn't be correct either. Part 2. A scientist is planning to test a new diet for weight loss. She selects 40 obese people to take part in the test. All the obese people are aged between 20 and 30. State 2 other factors the scientist should control when selecting the people. Anything sensible here? A similar body mass index would be a good idea. The same gender, so maybe just pick all females in one investigation. And lastly, make sure that they have the same lifestyle, so they carry out the same amount of exercise, because that would have quite a large effect. Devise a plan the scientists could use to test the effectiveness on the new diet using the 40 obese people. It's only worth 3 marks, so don't go too crazy on this. First of all, you want to start by weighing the 40 obese people. Ask 20 of them to follow the new diet, whilst 20 stick to their old diets, because after all, what they're consuming, i.e. the diet, is the independent variable. And then pick a sensible time frame, so maybe after 2 months, reweigh the 40 people. Gregor Mendel investigated the genetics of peas. He did not know about genes, but showed that inherited characteristics can be dominant or recessive. Explain how Mendel used homozygous tall and homozygous short pea plants to show that the tall allele is dominant to the short allele. So here you need an in-depth knowledge of how Mendel actually carried out his experiments. And notice that homozygous, let's pick T for our alleles. Homozygous tall would mean 2 big T's. Homozygous short would mean 2 small t's, and the only reason I know that is because I know that the tall is dominant to the short allele. So let's write what he did. So first of all, Mendel crossed homozygous tall and homozygous short pea plants. The fact that all the offspring were tall showed to him that the tall allele is dominant. And crucially, their genotype, if I draw a small Punnett square up here, so we've got a tall pea plant being crossed with a short one. Here's your Punnett square, so that's 100% would be tall, thus showing that the tall allele is dominant. Figure 4 shows a strawberry plant that has produced several runners and new strawberry plantlets are growing at the end of each runner. This is asexual reproduction. Explain why asexual reproduction in strawberries is beneficial to the strawberry farmers. So this is when you don't have pollination and that the strawberry plant can produce its own runners, and these are actually clones, so they are genetically identical to the parent plant. So why would this be good for the farmer? First of all, because it's extremely quick, and secondly, because there's no requirement for cross-pollination. It's a very quick process, and it allows the farmer to produce a high yield very fast. So why is it beneficial? Because asexual reproduction is a very quick reproduction technique as it does not require cross-pollination. Link it to the farmer's requirements. It allows the farmer to produce high yields very fast. Part 2. Strawberry fruits containing seeds are produced after a flower is fertilised. Explain why seed production is an advantage to the strawberry plant. So hopefully you know that in the production of seeds, sexual reproduction has taken place, meaning that 2 different strawberry plants have been used, and therefore there's genetic variation. We do not have clones here. And we know that genetic variation is key when the environment changes, because it means some of the strawberry plants will be better adapted. So let's write that. So sexual reproduction introduces genetic variation into the population. Which leads to an increased likelihood that a population will survive if the conditions change. And those are environmental conditions. So for example, drought, excess wind, too much rain. There will always be some plants which can survive based on the fact that they have genetic variation. Part C, Duchenne muscular dystrophy, I don't know if I'm pronouncing that right, is a sex-linked recessive genetic disorder caused by mutation on a single gene on the X chromosome. The letter D can be used for the dominant allele and the letter D, so lowercase, for the recessive allele. Figure 5 shows the inheritance of Duchenne muscular dystrophy in a family. Be careful, because it's a sex-linked recessive genetic disorder caused by mutation on the X chromosome, it means that D, so the allele which produces it, cannot be found on the Y chromosome. So if we actually have a look at the alleles, this genotype here would be for a healthy man, because remember men have an X and a Y. This genotype here would be for a sufferer of the disorder, and it again would be a man. If we now look at the female genotypes, this combination would produce a healthy female. The reason I know this is because it's a recessive disorder. This combination would produce a female with the genetic disorder, and lastly this combination would produce the healthy female. So these are the various combinations of alleles. So what is the percentage chance of any child from person A inheriting the mutated allele? So let's have a look at person A. Use the key. We can see that they are carrier females. Carrier means that they contain the allele but they don't actually suffer from the disease, which is why this must be their genotype. If we now look at B, because we're looking for a child, so we need to look at the man as well. Let's look at B. We know we have a normal male, and therefore to be a normal male they must have this particular genotype here. It's the only way they could be normal. With the smaller lowercase d, it would mean they would automatically have the disorder. So let's do a little Punnett square now, crossing the 2 highlighted genotypes. So there's the unaffected male being crossed with the carrier female, which means she has one big D, one small d. Do the cross, so that's a female offspring, so female child without the disease. This is a male offspring child without the disease. This is a female child that's a carrier that doesn't actually have the disease. And lastly, this is a male child that has the disease. So in terms of the percentage chance of any child inheriting the mutated allele, the mutated allele is this one here. So it appears in 2 of the children, so 2 out of 4 of the children have a chance of inheriting the mutated allele, which is 50%, which is why C is the answer. Explain the conclusion that can be made about the genotype of person C. So we're looking at this particular person. Genotype means we need to look at the alleles they have. Because they're female, obviously they have 2 X chromosomes. Because they're a carrier, it means they have one capital D, one small d. And so we can actually write that down. Question 5. Diffusion, active transport, and osmosis can be used to move substances into and out of cells. A student was investigating osmosis in potato cubes. He used the following method. Cut a potato into equal-sized cubes, record the mass of each potato cube, place each potato cube into different concentrations of salt solution, remove the potato cubes after 30 minutes, dry the potato cubes and record the final mass of each cube. He plots his results on a graph shown in figure 6. The method controls a number of variables. Name one other variable that needs to be controlled during the student's investigation. So as you can see, we're using different salt solution concentrations, and so that will be our independent variable. So everything else pretty much needs to be kept the same. That includes the mass of the potato chip, its surface area, the temperature, the type of potato, so any of those would do here. So I'm just going to write temperature here. Give a reason why the potato cube must be dried, and that's because you don't want any excess water on it, as that would give an inaccurate reading for the mass. Explain the conclusion that can be made about point Q on figure 6. So here, point Q, you can see that there is no change in mass because point Q sits on the origin, which means that there's no net increase or decrease in the potato chip's mass. Why is that? Well, that's because the concentration of salt within the potato must be the same as the surrounding solution. Hence, the potato didn't lose water or gain water. Give one way the student could obtain more data to increase the accuracy of point Q, clearly by taking more readings. So rather than just taking readings for 0, 0.2, 0.4, 0.6, etc., you could take intermediate readings. So you'd have one here, here, here. So repeat the test using intermediate concentrations. Osmosis is one method that single-celled organisms, such as bacteria, use to obtain molecules from their environment. Which of the following is a correct description of a process involving the transport of molecules? And so, as I look down, I can see we're comparing diffusion with active transport. Let's make a quick summary here of the two. So diffusion is the net movement of particles from an area of high concentration to an area of low concentration. This means that there's no energy required because it occurs along the concentration gradient. So it's a passive process. To summarize active transport, instead you're doing the exact opposite. So you're going from an area of low concentration to an area of high concentration. This obviously requires energy in the form of ATP, occurs against the concentration gradient. So let's hopefully find that one of these options agrees with something I just said. So, which of the following is a correct description of a process involving the transport of molecules? A. Diffusion is used to transport molecules against the concentration gradient. No, we've already said it's along the concentration gradient. B. Active transport is used to obtain molecules in a low concentration environment. So you should be able to see from my diagram that this is the low concentration environment and those particles are moving to a high concentration environment, which is why I think B is the answer. Let's check C. C. Active transport moves substances along the concentration gradient. No, I've said it's against the concentration gradient. D. Diffusion uses energy to transport molecules into cells. As you can see from my summary, I've said diffusion does not involve energy, which is why D is incorrect. C. Figure 7 shows some Vibrio chlori, the bacteria that causes cholera. The length of one flagellum on figure 7 is 68 millimeters. Calculate the length of flagellum in micrometers. You need this formula triangle, IM, which states if we're looking for the actual length of the flagellum, if I use my highlighter to cover A because that's the actual length, I can see the way I do that is by doing the image size divided by magnification. So let's write that formula here. It's really key here that we convert everything to the same unit, and because the answer is in micrometers, we need to convert millimeters to micrometers. The way in which we do that is by multiplying by 1,000. So when it comes to substituting our values, we've been told that the length of one flagellum on figure 7 is 68 millimeters. So we need to convert that to micrometers by multiplying by 1,000, divide by the magnification, which we've been told is 8,000. And when you pop that into your calculator, you effectively do 68,000 divided by 8,000 to get a value which is 8.5 micrometers. Six, DNA is composed of 4 different DNA nucleotides. Which diagram represents the arrangement of the sugar, phosphate and a base in a DNA nucleotide? Now this is something you're just going to have to learn, which is that the structure of a nucleotide is arranged in the following way, which is that it goes phosphate, sugar, followed by base. So this is simple fact recall. Really is worth learning this. And if you have a look using the key, you can see that the option which shows the phosphate followed by the sugar and then the base is B. An allele starts with a DNA sequence, A, T, blah, blah, blah. Give the sequence of the complementary DNA sequence and notice that it's the DNA sequence. Remember that adenine binds to thymine, cytosine binds to guanine, so the straight letters go together and the curly letters go together. So to find the complementary DNA sequence, I'm going to write it above. So that would be TACG, TACA, TGGC, and then just copy it straight down to avoid making any errors. The length of one DNA nucleotide was measured at 3.3 times 10 to the minus 10 meters. Calculate the approximate length of a gene containing 250 nucleotides in nanometers. Again, let's be careful with units. So we're converting meters to nanometers. The way we do that is by doing times 10 to the 9. So if we have 3.3 times 10 to the minus 10, convert that first into nanometers by doing times 10 to the 9, and then we've got a length of gene which contains 250 of these nucleotides. So that's why we're multiplying by 250. Once you pop that into your calculator, you get a value which is 82.5 nanometers. B. The DNA of an organism determines its phenotype. White tigers are produced because of a mutation of a single allele which usually produces the normal orange and yellow fur pigmentation. The mutated allele is recessive. Samba, a male white tiger, was bred with Rani. They had 3 offspring. 2 offspring have white fur, 1 has normal fur pigmentation. State the genotype of Rani. So genotype means the alleles that Rani must have. We know that Samba is a white tiger. So let's pick a couple of letters to act as alleles. So let's say big A, small a. We know that white coats are the mutated allele and that is recessive. So to have a white coat, the tiger must have this genotype. If they have capital A, or capital A, small a, then they'll be normal, so have the orange fur. Now, the fact that Samba is a male white tiger, this means that he has this genotype. He's bred with Rani and they have 3 offspring, 2 with white fur and 1 with normal fur pigmentation. Now, Rani must be heterozygous, so must be big A, small a, in order for them to have this offspring with both white fur and normal fur pigmentation. So we're going to say that Rani is heterozygous and that her genotype is big A, small a. The offspring with normal fur pigmentation, so that means the offspring of Rani and Samba, and therefore they must be big A, small a, because they can't be small a because they'd be white rather than having the normal fur pigmentation, and they can't be a big A because otherwise they wouldn't have had a brother or sister with white fur. So we know that the genotype of the offspring with normal fur pigmentation must be big A, small a, was bred with a tiger that was heterozygous, so actually the same, 2 different alleles. Use big A, small a to represent the alleles for fur pigmentation and predict using the Punnett square the percentage probability of the offspring from this cross having normal fur pigmentation. Nice and straightforward here. So we've got a big A combining with big A, small a, big A, small a, lastly small a, small a. So normal fur pigmentation is going to be these individuals. So that is 75%. Because 3 out of 4 of them would have the normal pigmentation. Explain how 2 parents with a dominant phenotype can produce offspring expressing a recessive characteristic, and this needs you to have quite an in-depth knowledge, and you need to know that both parents must be heterozygous for the recessive allele, meaning that the offspring must inherit the recessive allele from each parent. Figure 8 shows the diagram of the human eye. Which structure of the eye contains light receptor cells? The light receptor cells are the rods and cones, which are found on this portion of the eye around here, and we call that the retina. So option D is the correct answer here. The iris is the colourful portion of the eye, which is here. The lens refracts the light, that's here. The cornea also refracts the light, so it's here. So yeah, it is the retina which contains the light receptor cells. The optic nerve transfers electrical signals from the eye to the central nervous system. The optic nerve is therefore so try and remember for me that you go stimulus, receptor, and this is the order within a nervous response, to sensory neuron, to the CNS, to the motor neuron, and finally the effector, which brings about the response. So if we're transferring electrical signals from the eye to the central nervous system, the eye is obviously the receptor, so hopefully you can therefore see that the optic nerve is an example of a sensory neuron. So C is the correct answer here. Name the region of the central nervous system responsible for vision. This is again a factory call question, and it is the cerebrum. B. Macular degeneration is a defect of the eye that occurs when some cells of the retina begin to break down. Embryonic stem cell therapy has been used to improve the vision of some patients with macular degeneration. Explain how embryonic stem cells could be used to treat macular degeneration. So the great thing about embryonic stem cells is that they're totipotent, which means they can turn into any other type of cell. So we've been told that macular degeneration is when some cells of the retina begin to break down. So if we could use embryonic stem cells to produce brand new functioning retinal cells, then clearly we could help treat this disease. And once we get hold of these cells, they can be transplanted into the patient's eyes in order to help them see. So let's basically write that now. These can be transplanted into the patient's eye and can be used to replace the damaged cells. C. Some research has shown that increased use of computers and other digital media can affect eyesight and reaction times. The scientists wanted to test if prolonged use of a computer affected reaction time. The scientists tested the reaction times of 10 people under the same environmental conditions. These people then used a computer for 3 hours. The scientists tested their reaction time again. Give 3 ways that the scientists could improve this method to determine if prolonged use of computer affects reaction time. Right, let's do some highlighting to see what we're actually doing. So we've got 10 people here, so maybe use more people. They've said the same environmental conditions. Well, what are those? We've got a set amount of time, so that's 3 hours, that's pretty good. And the scientists tested their reaction time again. I think the key thing here, as with all experiments, is repeat, repeat, repeat. So that could be repeat with the same people or with additional people. So test more people. We said that they used the computer for 3 hours, but we could change this amount of time. So let's list some of these now. So change the amount of time that the computer is used for. Two, repeat the test using more people. And this note I made here about using the same environmental conditions, let's actually state that. So that could be the food and drink that the people have had before they take the test. Because if people have had lots of caffeine and coffee, that will obviously change their reaction time compared with people that have just eaten a huge roast dinner, for example, because I don't know about you, but I always get very sleepy when I've eaten a lot. D. Figure 9 shows the reaction times of 5 people, and this reaction time is given in seconds. Calculate the mean reaction time in milliseconds. Again, we have issues with units. To convert from seconds to milliseconds, we need to multiply these numbers by 1,000. And then the mean reaction time is obviously the average, so we're going to add up all their reaction times and then divide them by the number of people that there are. So I've multiplied all these reaction times by 1,000 to get them into milliseconds. And then we're going to divide the whole lot by 5. And once you've done that, you get a value which is 339 milliseconds. Give the name of the mathematical term which is used to describe the reaction time value of person 1. I don't really know how you're supposed to just know this, but if we were to arrange these numbers in order, starting from the smallest, so I'm going to do that here, you can see that 0.258, which belongs to person 1, is the middle value once these numbers are placed in order, so we call that the median term. 8. Phenolphthalein is an indicator. It is pink in alkaline solutions and turns colourless as the pH decreases. It can be used to measure the activity of the enzyme lipase on the breakdown of lipids. And if we actually have a look at lipids, remember under the action of lipase they turn into fatty acids and glycerol. It's really important you know this. Biology. Anyway, I digress. Samples of milk containing phenolphthalein were incubated with lipase at different temperatures. The time taken for the phenolphthalein to turn colourless was recorded and used to calculate the rate of enzyme activity. Explain why phenolphthalein turns colourless when lipase breaks down the lipids in milk. This is really key. Phenolphthalein is pink and alkaline and colourless when the pH decreases, and when pH decreases below 7 you've got an acidic solution. So the fact that the question tells us that it's gone colourless means that we have an acid from somewhere. Where could that have come from? Well, if you look up here at my notes, I've said that lipase catalyses the breakdown of lipids into fatty acids and glycerol, and hopefully you can see that word acid. So it's actually the fatty acids with their low pH is what causes the phenolphthalein to turn colourless. So let's write that explanation. So fatty acids are formed from the breakdown of lipids by the enzyme lipase. Fatty acids are acidic and therefore produce a low pH, which is responsible for turning phenolphthalein colourless. And I'm not writing that word out because that's so long. Describe the effect of temperature on the activity of lipase as shown in figure 10. So as always when you're describing you need to say what you see. So you can see that as the temperature increases from 20 degrees to 37 degrees, the rate of lipase activity increases. The rate of lipase activity is actually optimal, so at its highest rate at 37 degrees Celsius, and above 37 degrees Celsius, the rate decreases. So a nice straightforward describe question. Explain why the activity of lipase changes above a temperature of 40 degrees, and above 40 degrees you can see that you see a massive decrease in its activity. So it's really asking us, why does its activity decrease? And that's all to do with the enzyme being denatured, which means that the active site, so if this is your enzyme, when it gets denatured, this active site changes shape, meaning that the substrate molecule can no longer fit. And if you like these sort of perfect answers I'm giving, this is what my revision guide is full of. I spent all my time going through past papers and writing very concise notes for you guys. The student investigated the time taken for amylase to break down 10% starch solution into glucose at 37 degrees. The student repeated the investigation 5 times, and figure 11 shows the results. Calculate the rate of amylase enzyme activity for the 10% starch solution. So in order to work out the rate of reaction, you first of all need to find out this average time. So the way you do that is by adding these numbers together and then dividing them by the number of tests, so that's 5, to get a value which is 117.6, and then to work out the rate you just do 1 divided by that value. The investigation was done at 37 degrees Celsius. State one other variable that the student should have controlled during this investigation. So anything sensible here, from the concentration of the enzyme to the volume of the enzyme solution, the volume of the starch solution, and the starting pHs. But I'm just going to write here volume of starch solution. Different enzymes catalyze specific reactions. Explain why enzymes can only catalyze specific reactions. And as I drew very poorly above, it's all to do with that active site of the enzyme. It has a very specific shape that is only complementary to very specific substrates. That active site shape is dictated by the order of amino acids, because remember, enzymes are proteins, which means that they're made up of long chains of amino acids. Let's write that here then. So enzymes are proteins, meaning that they're made up of long chains of amino acids. This gives their active sites a very specific shape. Which only certain substrate molecules can fit into. Nine, when bacteria divide, they replicate their genome and synthesize their cell wall. Figure 12 outlines the stages of bacterial replication. So you have your cell wall, which contains the double-stranded DNA. The DNA gets copied to form the replicated genomes, and then you can see the cells splitting to form 2 new bacterial cells, which will be identical to the original cell. Penicillin inhibits the synthesis of the cell wall in bacteria. Explain the effect of penicillin on bacterial and human cells. Now, hopefully you know that because humans form part of the animal kingdom, and therefore their basic cell structure looks like this. It has the nucleus in the middle, surrounded by a cell membrane on the outside. There is no cell wall in an animal cell or in a human cell, and therefore penicillin will have no effect on human cells. Because human cells have no cell walls. However, penicillin obviously damages or prevents the synthesis of cell walls in bacteria, meaning that they won't be able to divide and won't be able to replicate in this way. B. Penicillin, isolated from a fungus, was the first antibiotic used to treat bacterial infections and is still widely used today. Scientists have genetically engineered bacteria to produce large amounts of penicillin. Describe how scientists would produce a genetically modified bacterium that produces penicillin. This is an answer that needs rote learning. You need to specify all the special enzymes which are involved and basically list a load of very key steps which are jam-packed full of scientific terms. So let's start by saying that you want to isolate the gene which produces penicillin and remove using a restriction enzyme. Next up, you want to use the same restriction enzyme to remove the plasmid of the bacterial cell. So the bacterial cell here contains both the nucleoids, so the DNA, and it also contains small circles called plasmids, and they're actually what's needed in genetic engineering, because what you do is you chop open those plasmids, you insert that gene that you're after, so the one responsible for producing penicillin, you pop it back into the bacterial cell reproduces and therefore produces the penicillin or the insulin or whatever you're after. So that's the summary, and we've already started writing the full answer. So use the same restriction enzyme to remove a plasmid from the bacterial cell. Next up, you want to insert the gene into the plasmid and use a different enzyme called a ligase enzyme to seal the plasmid. Now, when a plasmid has gained genetic information from another source, we call it a recombinant plasmid. So that's another key scientific word we're going to use. And we're going to say, insert the recombinant plasmid back into the bacteria. MRSA is a bacterium that has evolved to become resistant to antibiotics. With reference to Darwin's theory of evolution by natural selection, explain how MRSA bacteria have evolved to become resistant to antibiotics. Sounds awful. It is worth 6 marks. Again, if you've learnt this answer, it's actually a really nice question because it doesn't really rely on understanding and interpretation of questions and exam technique. It kind of just means you have to give a perfect answer that you hopefully learnt beforehand. So let's start with my perfect answer by saying that there is variation within the bacterial population due to mutation. It's always the first mark. This means that some bacteria will be resistant to antibiotics. So we're actually applying our perfect answer to the question being asked. And are more likely, therefore, to survive and reproduce. So these bacteria pass on these genes, these resistant genes, and soon the whole population becomes resistant to penicillin due to the process repeating over many generations. There are different types of white blood cell in the body. One type is called CD4+ T helper cells. The normal range of these cells in the blood is between 5 times 10 to the 8 and 1.2 times 10 to the 9 cells per decimeters cubed. An AIDS patient has a CD4+ T helper cell count of 1.5 times 10 to the 8 cells per dm cubed. So hopefully you can see much fewer cells in an AIDS patient. Explain why the CD4+ count of 1.5 times 10 to the 8 cells per decimeters cubed has led to the diagnosis of AIDS. Now, AIDS comes from the virus HIV, and HIV attacks white blood cells and actually destroys them. So this has been diagnosed previously by looking at AIDS sufferers' blood. So if you actually get a new person in and have a look at their white blood cell count and see that it is significantly lower, then it could really indicate that someone has AIDS. So let's make that comment first of all by stating what we can see, which is that this CD4 plus count is much lower than the normal range. And why is this? That's because the HIV virus has destroyed the white blood cells. Some sexually transmitted infections, STIs, can be diagnosed by testing urine samples. These tests use monoclonal antibodies that bind to an antigen on the pathogen. Describe how a monoclonal antibody can be developed and used to detect an STI using a urine sample. It is worth 6 marks, so we need to try and give 6 separate points here. And again, it's a type of method that is very much worth you getting to grips with and learning the procedure for. So let's first of all just point out a couple of these keywords. First of all, the antigen is the protein which exists that acts as a marker that enables you to recognize a particular substance. The pathogen is just a microorganism which causes disease. So in the first step of using monoclonal antibodies, you want to isolate an antigen from the pathogen which causes the particular STI. The next step when we're talking about monoclonal antibodies is we need to mention a mouse or any type of rodent which we inject the antigen into. The lymphocytes which are found within the mice produce antibodies which work against the STI antigen. So you want to harvest these, collect them basically. Our next stage is to produce the hybridoma, which is made by combining the B lymphocyte with a myeloma cell. And then the hybridoma's responsibility is to produce the monoclonal antibody against the antigen of the STI. In terms of its use, we want to attach this monoclonal antibody to a test strip, which can actually be used. Final question C. The antigens and pathogens can be proteins with a specific amino acid sequence. Explain how the order of amino acids in a protein is determined by the sequence of the gene. So really we're looking at protein synthesis here, and remember the 2 steps in protein synthesis are transcription and translation. Now transcription is all about producing mRNA, whereas translation is actually the manufacture of a protein on a ribosome. So let's first of all start by saying that the DNA in a nucleus unwinds and complementary mRNA nucleotides pair up. produce a single strand of mRNA. Protein synthesis can't actually take place within the nucleus, so the mRNA leaves the nucleus and binds to a ribosome. Now hopefully you know that in terms of reading the mRNA strand, let's create a small one here. It's called a triplet code, which means that the bases are read in threes. So a sequence of 3 bases is known as a codon, and it is complementary to a sequence of 3 bases which you find on a tRNA molecule which is attached to an amino acid. If you really hate what I'm saying here, you must watch my video on protein synthesis. It has it all in there. So we say that a triplet of bases So a triplet of 3 bases on mRNA is known as a codon, and it is complementary to the anticodon found on a tRNA molecule. And at the end of a tRNA molecule, you find an amino acid. So let's call this one alanine. That's the type of amino acid. Here's the anticodon, so it has 3 bases, and I'm going to match it with this codon here. So the complementary bases, therefore, are A, U, G. This ensures that the amino acids are assembled in the correct order on the ribosome.